And our next speaker is Anna Popova from Microarray in Karlsruhe. Anna received her undergraduate education at Lomonosov Moscow State University in Russia. She then moved to Germany, where she earned a PhD degree at the University of Heidelberg, working on new mechanisms of signals transduction in alternatively activated macrophages. She then took her maternity leave and started to do postdoctoral work sometime later on high-throughput screening in live cells at the Karlsruhe Institute of Technology, where, if I understand correctly, she's still working today after having, and take care of 2 kids at the same time. And at the same time, she co-founded Microarray in 2018, where she's the Chief Technology Officer. And this is a company that is concerned also with high-throughput screening technologies, the development of Droplet-Microarray technology for cell-based high-throughput screenings. And I guess this is what we're going to hear about, at least amongst other things, in your talk. So the screen is yours to share, Anna.
Well, thank you so much for such detailed introduction. It's nice to hear. I will share the screen now, and then I'll start. Just a moment. Yes. So can you see the screen? Yes, good. So hello everybody from my side. Again, thank you for the kind introduction and for the opportunity to present here. So the title of my talk today is Enabling Precision Oncology and Beyond with Novel Highly Miniaturized Chip Technology for Testing Live Cells. So I will start with very basic introduction in order to lead you through our vision, how we think personalized oncology can evolve in the future and how we can contribute to it with our technology. So we all know that cancer is a disorder characterized by uncontrollable overgrowth of usually one type of the cell of the body that afterwards spreads across all the organs and causes metastatic disease, which is usually the main cause of mortality in cancer patients. So cancer is considered to be an incurable disease and is the second leading cause of death worldwide. So now this disease is very heterogeneous and there are more than 200 different disease entities with survival rates ranging from almost 100% for testicular cancer and being almost fatal for pancreatic cancer. So the main line of therapy at the moment for cancer patients is surgical removal, radiation, and chemotherapy and targeted therapy. So the first two are focused on local removal or treatment of the tumor, respectively, when chemotherapy and targeted therapy are systematic treatment that introduced to the whole body. So chemotherapeutic drugs are usually generic toxic for all types of the cells, and with majority of drugs affecting the rapidly dividing cells, where targeted therapy is usually target specific biomarkers, usually a protein, that causes uncontrollable growth of cancer cells. So there are a few measures of precision oncology at the moment exist to identify the systemic therapy to which patient will most likely respond. So first is a targeted sequencing, which aims to identify the known mutation that can be treated with already existing targeted anti-cancer therapy. And this is well accepted measure in clinics. Along with that, it's possible to perform a full molecular profiling of a tumor with a goal to identify known variations in genome, epigenome, transcriptome, or metabolome of the patient present in the tumor in order to predict their response to our therapy. So pharmacogenetics is a branch of genetics which studies this kind of correlations between sensitivity or resistance of individual tumor to a drug and genetic background of a patient. So with the final goal to find the predictive biomarkers that can predict the response of the patients to a particular therapy. So now, despite all these advances, the problem is still persists that the most cancer patients do not respond to standardized treatments, including chemotherapy, targeted therapy, or even combined. So why is it so? First of all, the tumors do not always carry a mutation that can be targeted with existing therapy and not even known to be associated with certain drug response. In one of the studies, for example, it was shown that from sequencing of 3,000 different tumors, only a quarter contained known mutations that were previously even associated with the cancer. Second is the presence of known biomarker in the tumor doesn't necessarily guarantee a full response to a therapy targeting this biomarker, and this is probably due to cancer heterogeneity. It means that only some part of the cells might contain this biomarker and they will respond to therapy, while the other part of the cells would be resistant, contributing to further cancer progression. So as I said, cancer is very heterogeneous and it's not static. It's constantly evolving and changing throughout the tumor progression. And this is very important to think about it while adjusting the therapy for the patient. So in one of the studies where authors sequenced one particular breast cancer patient, they found that the number of mutations in this cancer exceeds 1,000, where 1/10 of such mutations were actually unique for this one particular cancer. And finally, our knowledge about cancer mechanisms and database of omic data is still limited, and we cannot with high probability predict their response to a therapy just based on different biomarkers. So what can be done to improve it? So the combination of molecular profiling data with ex vivo drug sensitivity and resistant test, where the biopsy cells of patient is tested ex vivo with a panel of anti-cancer drugs in order to find out to which drugs cells or tumor respond or resistant, can show the real-time responsiveness of the tumor. So combination of this sensitivity profiling and molecular profiling will contribute to understanding of a disease and biology of the disease and mechanism of a drug action. Also will contribute to ultimate finding of predictive biomarkers in the future, to predict the response of the patients to the therapy based on biomarkers. However, the main point of this test that the clinicians can see the response of the tumor in real time to different therapies in order to be able to find a suitable therapy for each particular cancer in this particular time point. So there are a few models for this ex vivo testing exists. So it's in vitro models, it's 2D cell culture models that used usually for blood cancer or for solid tumors where the cells are disintegrated and cultured in monolayer culture. It's 3D cell culture models by means of spheroids and organoids, and it's tumor tissue slices where the tumors are not even disintegrated but cultured and treated as small pieces of tumor. And this is the in vivo systems where tumor cells from patients are propagated in the model organisms such as mice or zebrafish. And in all these models, the treatment is being introduced, so anti-cancer drugs being introduced to the model with a final goal to define to which treatment this particular tumor is sensitive or resistant. So overall, such tests were shown before to be predictive of patients' response to a different therapy, and even 2D cell models alone were shown to predict the resistance in short-term response in patients with accuracy between 80 and 100%, which means if the tumor shows resistance to a drug in vitro with probability up to 100%, it will show resistance in patients. And it predicts drug sensitivity with an accuracy of 50 to 80%, which means that if tumor shows the sensitivity to a drug in vitro, it will, with probability up to 80%, will show sensitivity. So despite of these proofs, these tests are still not overall recommended, and it's not the mainstream treatment at the moment applied in clinics. So why is it so? The first reason is that there's simply not enough clinical studies at the moment being completed, which proves that such tests are predictive for short-term and long-term patient responsiveness to drugs. There are a number of clinical studies are in the progress at the moment. And the second reason is the platform reason, the protocol reason. So the most protocols that are being used for such tests that they're not compatible with high throughput. So they're not able to test large number of compounds and also combination of compounds. They usually require a large number of patient cells, which is very critical because patient material is limited. They're too long, So it's not, it's critical in order to have a decision on the therapy, and they're expensive, and they're not standardized. So there is no standard method which would be applied at least for one type of cancer, and each laboratory usually tackles the problem with different models. So what can be done to improve it and to accelerate the adaptation of this test to be incorporated in clinical practice? So when you imagine a platform which would be in the future used for routine testing of clinical samples that would be implemented in clinics, it should be miniaturized, so compatible with testing minimal number of cells. It should be high throughput and enable testing of large number of compounds, also combinations of compounds, and it has to be fast and able to deliver results within a few days in order for clinicians to start and make a decision on the therapy. So now I'm coming to our technology, which is called Droplet-Microarray. It consists of an array of hydrophilic spots on superhydrophobic background, and due to extreme difference in variabilities between these two areas, their aqueous solution forms the droplets spontaneously, as you can see on the video. So we can form an array of stable separated droplets of nanoliter volumes where we can culture and screen the cells. So when you look at the microtiter plate, you will have physical barriers between the wells to confine the liquid, and due to that, there is no possibility of further miniaturization beyond the microliter volumes due to physical and capillary effects. So in our arrays, we have an absolutely planar surface. There is no physical borders, and due to that, we can reach unlimited miniaturization down to nanoliter volumes. And this translates to 3 to 4 orders of magnitude less reagent consumption and up to 2 orders of magnitude less cell consumption. So these arrays are, as was already told, now commercially available through the company called Aquaray, and yesterday Sabrina Dellenbach from Geiger AG also have a nice talk about it. So what is all possible with Droplet-Microarray? So we can use it for 2-cell culture models. We used it for the past years with different types of cells with commonly used cell lines of adherent and suspension nature. We tried it with different types of cancer cells derived from patients, and we also use it for testing and screening of different types of stem cells. So this platform is also compatible with 3D cell models. Here we can use it in combination of the method of hanging droplet to promote self-assembly of cell spheroids or organoids, just simply by inverting the slide by culturing. We can also create different scaffolds or like hydrogels where cells can grow in 3D environment. And we also can test even small animal models like zebrafish, which ultimately can be used for testing xenograft models. So this is our workflow and protocol, how we use our arrays for drug screening. So first we preprint the library of anti-cancer compounds on the empty slide. Afterwards, we can store the slide, and whenever the patient samples is coming, we can use a low volume automated dispenser to dispense the cells and nanoliter droplets in each hydrophilic spots. And afterwards, all the compounds that were preprinted, they go into solution. And after 2 to 3 days of incubation, we can perform the readout. We can again use the automatic dispensing to dispense staining or fixation solution. And one of the way to do readout is to use microscopy. So Droplet-Microarray is compatible with all types of microscopy. We can do live imaging, staining, fixation, immunofluorescence, and all types of imaging. What else we developed lately is bulk colorimetric viability assays on Droplet-Microarray. Here we use 2 commercially available colorimetric viability assays, which are Resembles and VST8. And here you see the scans of the array. The scans of the array with containing HeLa cells treated with different concentration of the anti-cancer drug. So in the table you see the numbers corresponds to concentration, and here the color changed by live cells from transparent to yellow. So and you can see nicely that it corresponds where you have high concentration, there is no live cells, where you have a lower concentration, the color is changing, the same as here, it's changing from blue to pink. So we developed very simple readout where we can scan the slide using a simple paper scanner in one step, and we develop the pipelines and protocols to automatically analyze the depth of the color. So essentially we can assign the number to each spot. And we use this method to perform some dose responses using HeLa cells and number of anti-cancer compounds, and compared the microscopy-based readout that we usually use with two different assays. So we could obtain the similar dose responses and also the similar IC50 values, proving that this method can be used for primary screens for the high-throughput screenings. So we were thinking in the beginning if the cells actually respond the same to the drug in different volumes. So in the first part, we were testing, we made a simple experiment where we tested 100 cells in different volumes, ranging from 100 nanoliters to 100 microliters with a range of concentration of doxorubicin. So as here we showed that actually cells respond very similarly to the same concentrations, even though they were cultured in different volumes, and we also could obtain the similar IC50. So then we tested it and compared if we can get the same results on the chip and also in microtiter plates. So here there's example of HeLa cells that have been treated with a number of anti-cancer compounds, and we compared the dose responses in the plates and Droplet-Microarray. So as you can see here, we're getting similar dose responses, which was very encouraging for us to move on. So now coming to personalized medicine applications, for a couple of years, we working in close collaboration with a group of Sasha Dietrich in the University Hospital Heidelberg, where they perform such sensitivity tests or using blood cancer in 384 well plates. And we were miniaturizing this protocol on our arrays. So here we worked with chronic lymphocytic leukemia cells. This is the microscope image of them stained with calcium PI. We also developed the automated image analysis pipeline where we can automatically calculate viability in each spot. And here we can see comparison of viabilities of the cells incubated in 100 nanoliter droplets for 2 days, and also in flasks obtained from 5 different donors. So we went on and tested if we can get the same dose response from those cells on the chip compared to the usually used protocol. And here we compared the Droplet-Microarray using microscopy readout and 384 well plate using again microscopy readout, and the ATP-based viability assay, which is usually performed and used in these tests. So we got similar dose responses here, and we will continue collecting more information with more patients and also more drug entities. So we're also starting to use different cancer types. Here we work with multiple myeloma cells obtained from fresh biopsies of the patients. And this work fully performed in Heidelberg by Berit Brinkmann. And here is microscope images of multiple myeloma cells treated on our arrays with 2 different drugs and stained. So we use microscopy readout for this assays, and here we also use automated image analysis where we can estimate the number of viable cells, number of dead cells. So we continue this project of performing tests with more patients and with more drug entities to compare essentially our results in the well plates and Droplet-Microarray. And the first results looking very encouraging, that we can mostly see the same trend and the same effect using both platforms. But of course, with the Droplet-Microarray, we can use much less cells and also the compounds and medium. So to sum up, the Droplet-Microarray is quite versatile platform. It essentially can be used in microtiter plates. It can be used for screening of all types of cells, for 2D and 3D cell culture models, also for small animal models. It's compatible with standard readouts, with microscopy, and also bulk colorimetric assays, and also with standard equipment like microscopes or low volume dispensers. On the other hand, is much smaller, so we can achieve 3 orders of magnitude less cultural volumes, which translates in the requirement of much lower cell numbers, which is very critical for such applications. And also we can achieve higher throughputs compared to microtiter plates, ultimately enabling screening of more compounds. So our future vision for this in vitro drug sensitivity and resistance test is, first of all, it should be adopted for all or most types of the cancer. It should be a physiologically relevant in vitro model that is appropriate for each type of cancer. It can be 2D or 3D. Of course, it should be implemented in the clinics, and it should be standard protocol for one type of cancer. So if all of this in place and this test is working in clinics and implemented, it will allow us, first of all, to monitor the disease progression and clonal evolution, meaning that it would be possible to take the samples before, during, and after the treatment to see if the cells that showed resistance or sensitivity before treatment is actually still present, or there's other cells evolving that can be resistant to a therapy that actually in the moment introduced. So this will allow to adjust the therapy in the real time. Also, such tests will allow repurposing of drugs. If we're able to test larger number of compounds, also combinations of compounds, it's possible to find a drug that would be effective that usually are not used for this type of cancer, also broadening the option for the patients. It would be possible to monitor metastatic cancer, to get the biopsies from metastatic sites and even circulating tumor cells, which are shown to be quite different, especially in the drug response compared to original tumor. And last but not the least, this technology will allow, this test will allow mining a new fundamental data. So finding the relationship between drug response and also different omic profiling data of bulk tumor before the treatment, after the treatment, but also the possible alteration of epigenome or transcriptome after the drug treatment on the chip, allowing completely new data we can get, and with ultimate goal to first understand more the mechanism of disease progression, the mechanism of drug treatment, but also finding the biomarkers that potentially can be predictive of their response to therapy. So that's our vision, and I'm by the end of my talk. So I would like to thank my wonderful group, our collaborators, our sponsors, and thank you very much for your kind attention.